Hello, my name is Emily Linkevich, and I am an undergraduate at Albright College. This summer, I did a continuation of my project from the previous year in which I looked at the effects of the non-native Microstegium Vimineum on small mammals and insects. I would like to first go over three of the major components of this project, the first being Peromyscus leucopus. Most commonly known as the white-footed mouse, these mice are habitat generalists in which they eat a variety of foods and thrive in a wide range of habitats, though the white-footed mouse is primarily a woodland rodent. They feed on insects, fruits, and seeds. However, arthropods are the most commonly identified item within their stomachs. The second component is Microstegium vimineum, commonly known as Japanese stiltgrass or packing grass. The species is native to Japan, China, India, and Central Asia. The first documentation of this species in the United States was in 1919 in the state of Tennessee. Since then, it has slowly made its way up the eastern coast and has been observed in Nolde Forest on the trapping grids and sites, in which trapping began in 2003. The plant looks like thin, wiry bamboo and has a silvery stripe on the midrib of the leaves. This species is also shade tolerant, meaning it can live in low light levels, which help it thrive in a closed canopy forest like that of Nolde Park. The last major component is that of the Magicicada, commonly known as the periodical cicada. In the summer of 2021, brood 10 emerged, which includes three different species, Magicicada septundecim, Magicicada cassini, and Magicicada septidecula. They tend to live for 2 to 6 weeks after emerging. The chorus can reach up to 100 decibels, which is comparable to a jet flying overhead at 1,000 feet, which equates to 103 decibels. The emergence of the periodical cicada is considered to be an abundant food source for that of small mammals. As this is a continuation, I'd like to include two of the results from last year. First being small mammals were more likely to be impacted by stiltgrass than that of insect diversity. Along with that, the analysis found that insect data was not sufficient enough to find an impact from Microstegium vimineum. However, this could also be due to a little under half of our insect data being unidentifiable, as they were in pieces, or I could not figure out even with the help of a dichotomous key. Next is the objective of the study, which was to gather more insect data to determine whether stiltgrass affects insect diversity and abundance along with the activity of small mammals. Another key point was to determine whether the emergence of the periodical cicada affects the insect abundance and if the mouse population will increase. Previous literature, Dr. Meck and Chase in 2004 studied the Nolde Forest Park and found that the site in which they were looking at during the last periodical cicada emergence and found that the population increased, therefore leading us to a prediction that the mice population will greatly increase in density. Next, Medikoff and Emery, there has been an increase in insect populations when Japanese stiltgrass is present while the native populations decrease. A higher density of cicadas tends to result in a higher abundance of reproductive females. And this last point is a bit contentious, but I found it interesting nonetheless. There is a possibility that the mice can anticipate the periodical cicada emergence, therefore increasing their reproductive status before the emergence. However, there's a lack of data for this. For 2021, the data collection was from April 17th to July 2nd. For 2003 and 2004, only the select cut site was used, which is an 8x8 grid, but for 2020 and 2021, the clear cut control site was also used, which is a 5x5 grid. The location of the study was at Nolde Forest Park, which is just outside of Reading, Pennsylvania. For the mice, we set up baited traps with rolled oats and peanut butter and bird seed. The traps were opened on Tuesday afternoon and checked twice a day, Wednesday and Thursday, and closed Friday morning. This was to reduce the amount of stress on the animals. The data recorded for the mice included pit tag ID, sex, age, reproductive status, and ectoparasites including ticks, flea, and bots. and any other notes of interest. Though the insect data was not officially recorded in 2004, it was the first time of note in which the periodical cicadas emerged. The second time in which the insects were noted was in 2020, in which we actually collected insect data using pitfall traps and sticky traps. In 2021, we only used tanglefoot traps to capture the insects. For this, we used 38 on the select cut site and 32 in the clear cut control. For this, the selection of the traps, we used a random number generator to pick the locations of the traps. After setting them out at the same time the traps were set and bringing them back in once the traps were closed for the week, they were brought back to lab. Using a dichotomous key based on the taxonomic level of order, we identified the insect with the help of a microscope. Results for the data analysis of Peromyscus leucopus population. In 2004, it had the highest population rate. In period 1, there were a total of 144.7 individuals during the period. In 2021, we did not find a similar increase. However, it was relatively similar to the results of 2003 and 2020. The total population for 2021 for the select cut site was 96.7 individuals, while on the clear cut control site, it was 33.3 individuals. Results of the data analysis for Peromyscus leucopus survivorship. The survivorship is the probability of the population surviving each week. The population survivorship was relatively high throughout all the periods, around 70%, indicating a favorable environment. Data analysis results for Peromyscus leucopus births. The births were the number of individuals that were new each week. There was no consistent pattern for the births, and in 2004, the highest births were 20.4 individuals, while in 2020 had the second highest births at 18.15 individuals. Overall, the mice data appears to be inconsistent with our predictions. We predicted that 2021 would have a similar increase as 2004. However, as previously stated, the data for 2021 showed the population was relatively similar to 2003 and 2020. Speculatively, the increase in 2004 could be due to the masting of oaks, in which masting is when the oaks in a region release all of their acorns at once, leading to an overabundance of food. This, however, did not occur in 2021, though previous literature did suggest higher densities of periodical cicadas leads to a higher abundance of reproductive females. Our results for the births found the rates vary between each emergence. The future of this project is I am currently working on it as an independent study focusing on the insect and vegetation data. I also hope to present at the Pennsylvania Academy of Science or at the annual meeting of the American Society of Mammalogists in Tucson, Arizona next year. I would first like to thank the Albright Creative Research Experience, or ACRE, and the Hearst Foundation for funding this project, Dr. Meck for being my mentor and usually having to wait until I give up and ask for help on the project, the Nolde Forest Environmental Education Center, and the Pennsylvania Department of Conservation and Natural Resources for access to the site, Ryan Brett and Bio 383, or Population Ecology, for help in collecting the mice data, and Dr. Osgood and Abigail Shoemaker for helping collect the vegetation data.